In this video, we will discuss about the molecular phenotype guided therapy in pancreatic cancer. In pancreatic cancer, the success rate is pretty low. Success rate is low. This is because of the complexity or different drivers results in the activation or development of tumor within the pancreatic cancer. Some of the though the mutations in the pancreatic cancer is low, still people have not been able to find out a more efficient way to cure pancreatic cancer. Some of the genotypes or some of the drivers mutation has been studied, and they have also been studied independently, which in total accounts for around 45% of the mutation, which account 45% of the mutation that leads to pancreatic cancer. And several therapies has been tested on the pancreatic cancer. One of them is like platinum based therapy. So what happens in this case of platinum based therapy is highly hypersensitive to DNA damage repair. Hypersensitive to DNA damage. So people have identified the factor that causes this DNA damage. It's something called, like say it is XYZ. You can find that exact name in the reference paper that is available, or the link of that reference paper is available in the description. So this gene might lead to hypersensitive reaction, or is hypersensitive to DNA damage factors. So what people have think that if this combination has been removed from this platinum, or this things can be inactivated, or this gene can be inactivated or removed from the platinum based theory, then efficiency of this platinum based theory could be improved, and this problem can be minimized to much bigger extent. And with the advancements of the technology such as genome wide sequencing, people have divided this pancreatic cancer into 4 major categories. One is stable, one is localized, one is scattered, and one is unstable, or the cancer that follows the pathway BRAC pathway. And this is where most of the problem occurs, as people have not been able to identify the drivers mutation that leads to pancreatic cancer. And this thing BRA pathway is mainly related to DNA damage. DNA damage. What people have think that whether this unstable pathway also include the things that does not have DNA damage or this bark pathway, and people have find out that 14% based on the study, they have find out that 14% of the cases, 14% of tumor does not have this BRCA pathway, or 14% of the tumor are non-BRCA pathway. I think it is BRCA2. We can confirm this from the reference paper. Non-BRCA pathway. So this gives a positive indication that genome-wide sequencing will take into account even those pathways that are not yet been explored in terms of drivers for pancreatic cancer. So this is just a small overview what is at present going around for the treatment of pancreatic cancer. So that's all for this video. Thank you for your time.